Welcome everyone to this first Prime workshop, and I'm going to hand you over to Sjoerd Bentjes, who is going to chair the first session.
Thank you very much, Chris. Hello everyone. My name is Sjoerd Bentjes. I'm a lecturer in mathematics at the University of Edinburgh. And as Chris said, welcome today to this first Prime workshop titled How AI ML Methods Can Enhance ME/CFS Molecular or Genetic Biomarker Discovery. The aim today is hopefully that together we can drive forward science in ME/CFS and in long COVID by presenting state-of-the-art AI and machine learning and statistical methods for application in this domain. And a number of key considerations I've written down here for you today. So the first is what we aim to have a focus on clarity. So what is the scientific question of interest? What is the data that we're working with? There'll also be an emphasis on rigor. So which method is chosen to answer the scientific question and why? Are the assumptions of the method valid, or should something else be used? And thirdly, a focus on uncertainty quantification. So even if we get some nice results out, how certain are we of those results? Throughout, we'll try to make a distinction between two main areas of analysis, estimation and prediction. So for estimation, you can think of a population average statement. Say there's a group of individuals with ME/CFS where a biomarker has been measured, and a group of healthy controls where a similar biomarker is measured. You can ask what the difference is between these biomarkers on average. So this is really a statement about the population. And in contrast, prediction is often an individual level statement where for a given individual, say from a group of biomarkers, we try to predict the odds of them developing ME/CFS. So one of the things we're very much aware of is that today is an open session. So everyone is welcome, which means that there are attendees and speakers with varied backgrounds. So we've done this on purpose because we feel insights often emerge from exchange, from crossing disciplines, and from working in between disciplines. So today we've asked the speakers to prepare a talk of about 20 minutes so that we leave 10 minutes for questions. So if you have questions, please type those in the chat. We'll select a few of those to discuss during the 10 minutes, but we'll also try to address a number of them during the talks themselves. So speakers again have been asked to comment on the methods they use to contrast valid application versus common mistakes and to exemplify applications to ME/CFS and long COVID, just as the three sort of aims that I mentioned on the previous slide. Now, before I introduce our first speaker, I first want to hand over to Chris Ponting, who will say a few words about the Prime Network more generally. So Chris.
Thank you, Sjoerd. So this is the first of 16 workshops under a new network called Prime in the UK, and you can see that Prime stands for Infrastructure for Patients, Research and Industry for ME/CFS. And the logo comes from someone with lived experience of ME, and I particularly like it because the M and E are kind of indicating the often invisibility of people with ME. So we will travel quite a distance over the 16 different workshops. We will come back to machine learning and AI. For example, in a later workshop, we will have Precision Life talking about their recent results. Next slide. So Prime itself has some large aims. We want to build a solid foundation for a future and permanent enabling infrastructure for ME research. And you all will know that there has been decades of underinvestment and often unreplicated research findings. And we want to reverse that. And so we want to accelerate towards effective diagnosis and treatment. as we all do. Yes, next. So we have three aims. These are sort of uh, upon which it will be determined whether or not Prime has been successful. That we will initiate or help to initiate novel collaborations, about 15 or more, with scientists, with industry, etc., and to catalyze over £1 million in funding over the 4 years of the project. We want to also catalyze connections between different disciplines and across different countries. So we want to catalyze an international genetics consortium and an omics consortium. And so that will allow analysis across different data and often across different ancestries to ensure inclusion and relevancy. And a third aim, which is incredibly exciting and led by Action for ME, who is, of course, a key partner in Prime, is to establish a 100-strong expert pool of PPI members who will be ready and waiting to be involved in new ME research in the UK. So this is a long-term resource for the research community. So I think that's me done. Let me make sure that is the case. Yes, there we go. So if you want any more information about Prime, then please can you contact Ed Hutchinson, who's the project manager. I'll put in his contact details in a minute. We have the academic leads named there. Daphne from Action for ME is key individual. And as always, we have involvement and our PPI team, who is incredibly important for us. The whole project is run out of the ME Genetic Centre of Excellence between ourselves here in the University of Edinburgh and Action for ME, and it's funded by the UK Medical Research Council. Thank you, Stuart. Enough from me. Onward we go.
Right, thank you, Chris. So now it's time for our first speaker today. So I'll stop sharing screen so that Nima can take over while I give a short introduction. So Nima Hijazi is an assistant professor at the Harvard Chan School of Public Health. So we have, full disclosure, worked together on a project on ME. So we're very happy that he's here today. So his interests lie in the development of assumption lean methods in statistical causal inference that are often driven by specific questions that come from application. And a second interest is in the development of open source software to enhance reproducibility and reliability of results, which is ever so important in this age of big data. So Nima, over to you.
Sorry, I forgot how to unmute for a second. Thank you for the introduction. So yeah, I'm excited to be able to talk about some work today on that stemmed out of a collaboration on long COVID, and that I think will have some parallels to work in MECFS. So I'd like to just thank the organizers for the introduction and note that this work was really joint with the team at Massachusetts General Hospital that included Andrea Folks, Harrison Reeder, and Wei-Shin Kwong. Yeah, okay, whoops, let's try that again. All right, so I'll start with a very brief introduction of the motivating study, which was called RECOVER, and that's Researching COVID to Enhance Recovery. This was an initiative created by the US NIH around 2021 or 2022 specifically to study long COVID in the US and to characterize the condition. So it was a large-scale observational cohort study with about 15,000 adults enrolled across the US. And it included two forms of enrollment. There was prospective follow-up from the date of the study's initiation. So individuals with acute SARS-CoV-2 infection could enroll, and also individuals who were uninfected could enroll, and infection was judged by individual report. And then there was a retrospective, enrollment component where individuals could contact the study team and note that they had an infection sometime in the past, and be enrolled in that way. As it says here, it includes infected and uninfected participants, and included quarterly symptom assessments. It also included clinical visits to specific clinics, and assessment of a wide variety of symptoms to characterize different forms of long COVID. So there's a lot of literature produced by Recover that you can look into if you're interested in that area. But it is a condition with kind of myriad symptomology, and so relates to ME/CFS in that way. Key objectives of Recover were to characterize long COVID, to define clinical subphenotypes, which they've now done, as well as trajectories of recovery, and then to look at the biological mechanisms of pathogenesis. And I'll focus on this last one here. So importantly, the study involved a tiered testing strategy. So individuals who were enrolled were sampled to collect further information based on how they were performing in the study. And by that, I mean whether they exhibited particular symptoms at specific times, whether their lab results were indicative of something that would have benefited from further study, their infection history, things like this. And there were 3 tiers. You'll see that tier 1 is basically all the individuals. And this is just surveys, basic labs, collection of biospecimens. Tier 2, which is collection of what we're terming low-risk clinical assessments, where things like liver elastography, pulmonary function testing, things of that nature, which were somewhat so these are sort of mid-cost kind of assessments. And then tier 3 were invasive, were things that were invasive testing. So these are like brain MRIs, for example. And you'll see that the number of individuals decreases quite a lot as you go through the tiers. So this is in part a resource allocation problem, and also importantly, one of the goals is to make sure that participants in the study are only undergoing assessments that are necessary for them to benefit from a health science perspective. So you don't want to give someone an MRI that doesn't need it, right? Okay, so with this design in mind, I'll discuss some statistical methods that are necessary for the recovery of population level estimates, despite this kind of selection bias that's baked into the design. Okay, so we'll jump to some notation now. I'll start with what the problem could look like if we didn't have this sampling structure, and then I'll layer on the sampling complexities as we go. So just to formalize a bit, we'll consider some study like Recover that had n study units or participants. The data on a given participant will represent as O for observed, and it'll have a couple of components. Those will be L, A, and Y. L is going to refer to covariates and comorbidities that are measured at enrollment. So these are things like sex at birth, age, race, ethnicity, alcohol use, vaccination status. That's important, right, for SARS-CoV-2. A is an indicator of COVID-19 infection. In some parts of Recover later on in the study, some of the questions are about the impact of long COVID or not at a given time point on some other health outcome. And so you can redefine A to be long COVID at a given time if you'd like. And then Y, in our example here, will be a vector of candidate biomarkers. So these will be things that biological measures that could be related to SARS-CoV-2 infection, and that may be on the pathway between infection and long COVID, for example. And these are going to be things that you could sequence from biospecimens, but maybe you don't want to sequence it on all the individuals. What we'd like to learn ultimately is an average treatment effect, or ATE. So this is for a given biomarker, call it little b. This is going to be the difference in the biomarker outcome, Y sub b, between individuals who are, if everybody were infected versus if nobody were infected. So how does the question this answers is, how does the expression of this biomarker be? How does this expression profile change as a consequence of infection? In causal inference, one formalism for expressing this is a potential outcome. So we'll denote that as y sub b for the outcome of the bth biomarker, and then super A for infection status A, little a. So this could be y sub b of 1 is going to be the biomarker profile under infection. Y sub b of 0 is the biomarker profile without infection. This is, importantly, this is a counterfactual statement. So it's for a given individual. I'm saying, what would this biomarker have looked like under infection or not? We see one of these things depending on what happened in the world. And then the ATE carries interpretation as the causal impact of infection status on the expression or activity of that biomarker. Of course, if we could randomize participants or patients to infection status, we could learn this ATE without too much trouble. And this is because randomization gets rid of baseline confounding. Of course, we can't randomize individuals to infection status, so we'll have to deal with some complexities. One framework that's become popular in epidemiology for making sure that we align observational studies with randomized trials is target trial emulation. So this is the practice of taking an observational study and mapping it to criteria that you would use in designing a randomized controlled trial or RCT. So the exercise is to imagine a hypothetical, or in the language of this literature, a target trial, and then imagine what it would look like to randomize participants' infection status. So some key criteria that were outlined by Hernan and collaborators who developed this framework include things like who would be eligible for this so-called target trial, what are the treatment strategies, treatment in quotes, right? We're talking about infection status. But this is helpful to think about what are we contrasting? How would this treatment be assigned? When does follow-up begin and end? This last bullet is actually what I often find to be the last and first bullet, I should say, are the things that I find to be the most useful, because there's often it's difficult to find clarity in the space between those that designed the study and then the statisticians analyzing it. So I don't often know what the index time should be, but when we sit down in a room together, the applied scientists and I, we can figure out these important things and make sure that we're on the same page as to what we're estimating. Okay, so this is just an example of what going through this exercise looks like. That very leftmost column is just the various components, and then the middle column is this idealized RCT or target trial. And then the rightmost is the emulated trial. So this is what the observational study that mirrors this target trial, what that looks like. So the aim, I won't read these out, but the point is we go through what is the aim, what is the eligibility, what are the treatment strategies, et cetera. And you go all the way from stating the aim down to the causal contrast is the penultimate component, and then what is the statistical analysis that will recover that causal contrast. So having done this, we now recover adult, that cohort of 15,000 individuals in this top box, exclude some individuals, look at individuals with some amount of follow-up from index, split them into infection status, and then get to the bottom boxes, which are those individuals for which we observe an outcome and can then use for our analysis. So we went from 15,000 to about 11,000 individuals in going through this exercise. Okay, so now we have our data set, and we'd like to think about how do we estimate this causal estimand, this average treatment effect. So we need at minimum some identification assumptions. These are two prominent ones are exchangeability, which is this statement here is that the biomarker outcome, the potential outcome, is independent of infection status given baseline covariates. So this is to say that conditional on the baseline covariates, it's as if infection status is randomized. And then positivity is that in every stratum defined by the covariates L, there has to be some positive probability of infection. So if there exists some stratum that could not be infected or could not be uninfected, you won't be able to learn from the data what you need to do, what you need to learn to be able to estimate this quantity. Applying these assumptions, you can take the causal estimand that I had a couple of slides ago and rewrite it as a plugin, a statistical estimand. That's the PI for plugin. So this is now, if we stare at this object for a moment, starting with the inside, we'll see that it's the conditional mean of the outcome for the Bth biomarker under some contrast of A is 1 or A is 0, depending on which term you're looking at, conditional on L. And then the outer expectation marginalizes over the covariate distribution. So what it's telling us is we need to estimate the outcome, the conditional mean of the outcome given infection and covariates, predict what that outcome would have looked like, the conditional mean would have looked like under A is 1 and A is 0, and then marginalize out the covariate distribution. And that becomes just a simple finite sample mean in practice. Another representation is this inverse probability weighted or IPW representation. So this writes things in terms of the probability of being infected. I'll call that G0. And you can see here now we have just YB kind of off to the side, and then a contrast of were you infected, that's the indicator A is 1, over the probability of being infected, and then minus were you uninfected over the probability of being uninfected. These are equivalent estimands arrived at from the same more or less identification exercise. So you can use either of these to estimate that average treatment effect. This would be fine if all we had was this observational study where we had possible confounding by L, but none of the sampling complications. So instead, we have some other issues that come up, and the data structure that I told you about is not, in fact, the data structure that we have to deal with. The observed data unit O still has L and A, which were the covariates and the exposure respectively, but then it also has some other components, namely T, which are labs that trigger further testing, R, which is an indicator of selection by the tiered sampling process, C, which is an indicator of censoring or non-completion. And then that last component, CRY, is saying that we will see the biomarker outcome Y if you are uncensored, C is 1, and if you're selected by the sampling mechanism, so that R is 1. So we only see Y for that subset of individuals. I think that's all this slide is saying. We have a subset that is impacted by at least 2 forms of selection bias, which are from the sampling that's in there by design, and then completion, which is at the discretion of the participants. And then this tiered sampling design was also constructed. This is actually the tier 2 mechanism, but it was constructed in a way that oversamples individuals who could benefit from further testing. So what this is saying is if an individual was infected and they activate a trigger, they are sampled with 100% probability. Otherwise, they have a 5% probability if they're infected and fail to activate, and if they're uninfected, they have a 11% probability. So you're going to get at the end of the day, a pool of individuals who are that overemphasizes individuals that are infected and have a trigger. The purpose here is to target resources to participants who could benefit from the testing and personnel time, and then to reduce cost by not sequencing as many biomarker assays as you normally would if you tried to do this on everyone. Yeah, so this is the design, and now we're going to just briefly go over. Hopefully, I have 5 minutes or so to go over how we'll correct for this design. And the way we do that is going to turn out to be just to use inverse probability of sampling weights. I say just, but because the IPW estimator that you just saw, if this was familiar to you before, then it's sort of a review, but otherwise you see that we use these inverse weights of the infection probability to account for confounding. And now we're gonna use that same principle to correct for the sampling. So we take that initial IPW estimator that we had, and we add an extra term that's now the indicator of where you sampled. That's the numerator. And then the probability of being sampled given your triggers, your exposure status, and your covariate history. And this new IPW estimator now it addresses this imbalance in the study, in the sampling design. We can achieve something similar with the plugin estimator. This is based on this so-called outcome regression, which is what I referred to earlier. It's the conditional mean of that biomarker YB under some exposure contrast little a and covariates L. Here, you can either fit this with inverse probability of sampling weights in the regression and generate counterfactual predictions, or you can fit it on the subset of individuals who complete the sampling process or who complete who are sampled, sorry, with R as one. Depending on the assumption you use, you may or may not need the weights. Okay. So what's the story if there's censoring? So this is when individuals who were sampled don't necessarily complete. So maybe you're somebody who was infected and had a trigger, and at your clinic visit you're told, hey, you've been sampled. It would be great if you could come in for further testing, but maybe you're unable to for a variety of reasons. And that C is 1 indicates that you did complete it. C is 0 indicates that you did not complete it. This is a form of selection bias that just arises naturally. It's not part of the study design, but we still need to address it. And you can see that the IPW estimator is identical to the previous one. We just now replace the C's with R's. If you were to rewrite this probability in the denominator as the probability C is 1 given A is L, A and L. So if it's not dependent on the triggers, you can simplify the outcome regression, which follows there. So here I say that you can fit it only on participants who complete the assay, and this is true if what you need for the particular missing at random assumption that you make, if it's only dependent on A and L, that holds. So more or less it's the same game as with the sampling, and then we can do them together. So now I've just put them both together. This would recover this would get back the population level average treatment effect of infection on the biomarker outcome in the full recover adult study population, despite both sampling and censoring. Importantly, though, these estimators are only valid if the nuisance functions are fit with restrictive parametric models. Part of the goal of this session is to talk about machine learning. So we've built up some machinery, but the machinery doesn't really allow the use of machine learning. Turns out that there are results. I would encourage you to not really stare at this equation, but I'll point out terms that are familiar. If hopefully my cursor is coming through, you can see here that there are the weights from the IPW estimator that we looked at. There are the same censoring weights and sampling weights that we looked at. And then there's terms from the outcome regression. And then there's some other stuff. The form of this equation is kind of, it comes from an area called semi-parametric theory in statistics. The function phi itself is called an efficient influence function. And it's been studied as early as the 80s. But its role in our story here is that it informs the construction of estimators that will allow you to use machine learning to estimate nuisance quantities, by which I mean this G0 of L and this M0 of 1L, those things that we need to estimate that are not our objects of interest. And importantly, the efficient influence function gives the asymptotic variance, the efficiency bound for estimators in a broad statistical model, and then allows the use of machine learning. So how can we use this? I'll probably just stop after this slide to say that there are at least two estimators you can construct based on that complicated looking equation. One is this one-step estimator, which takes the plugin estimator that we saw before. I've replaced the zero subscripts with n's here just to indicate we're now doing the estimation. N is for empirical. And then we're going to add to it this mean of the efficient influence function. So we're going to estimate this quantity, take its mean, and then add it to the plugin estimator. That then allows us to use machine learning for the estimation of the nuisance functions, and then still recover the average treatment effect. Another alternative is a targeted minimum loss or a targeted maximum likelihood estimator, which looks just like the plugin estimator, but I've added these stars. And the stars just indicate that we're going to take the initial outcome regression and then update it in a way that satisfies that efficient influence function equation. So this will be a plugin estimator that allows the use of machine learning and gives you the best possible variance in this broad class of estimators, by which I mean you'll get the tightest confidence intervals and the most sensitive hypothesis tests. So you can use arbitrary machine learning, and these are doubly robust, so they'll still be correct if one or the other of the nuisance functions is incorrectly estimated. I'll skip this simulation and just say this kind of tiered sampling strategy, which conserves resources, which may come up in observational studies of ME/CFS and certainly does in long COVID, requires analytic corrections. The tiered sampling helps us avoid needlessly testing study participants. So that large portion of individuals without triggers, it conserves resources, it saves patients the trouble of participants, the trouble of coming in for tests that are unnecessary. Unfortunately, this as a consequence of this sampling, we need to correct for it analytically, which we can do, but it makes the estimators more complicated. And the estimators that we've outlined here are really just a flexible, robust, and efficient way of using machine learning while addressing issues that are really causal inference issues, a lack of randomization, bias sampling, selection bias from censoring, these sorts of things. And I think I'll just stop there. Yeah, thank you.
Great. Thank you very much, Nima. Yeah, so I think the general tendency you mentioned here, so the approach using tiered sampling, where not everyone needs to be necessarily tested, is probably a very welcome strategy, both in long COVID and in the area of ME/CFS. So that sounds very interesting. So there are a number of questions here in the chat in that regard. So let me read out the first one. So you mentioned that there is informative missingness by design, essentially because of the tiered sampling, where not everyone is measured. And you integrated this information as part of the estimation procedure. Too often we see research papers using instead naive imputation to deal with missing data by just filling this in, or simply remove samples that have missing data. So could you please share your thoughts on the importance of taking the approach you took today, correcting for it, and not using naive imputation or removal?
Sure. Yeah, this is an excellent question. Thank you. So I'll start with the more naive approach of doing a complete case analysis. So when you drop individuals so I'll just maybe go back. When you drop individuals who go back to this estimator who maybe there's some missingness for them, and you don't implement a correction based on inverse weighting or based on imputation, and I'll come back to that second one in a moment. What you will get is inference for the wrong group of individuals, right? So here I say that the inference that we get ultimately is about the recover adult study population. If you don't do this, if you do a complete case analysis, it's for that group of individuals that happen to be able to complete the test. And so the conclusion that you would like to draw is not supported by that analysis, where the conclusion is usually something about the full study population. That kind of complete case analysis, frankly, should not be done unless yeah, I can't really think of a reason that you would do that when you know that this kind of selection bias is present. So that unfortunately, I agree it happens in practice, but we should move towards a standard for analytic methods that avoids that. With the question about the second part of the question about imputation methods, what I wanted to note is just that this plugin estimator, it does a kind of imputation. Maybe I'll go back to just here where I note that you can fit this outcome regression only on participants who complete, and then get counterfactual predictions. And so this is saying we're gonna fit on those individuals who complete and then predict on everyone. That's an imputation that's happening. It's correct under an assumption that the censoring process is independent of the outcome given what you've already adjusted for in this model, which is A and L. So if in fact the censoring process also depends on T, then what I've written here is incorrect. So it takes some care to figure out when you can fit these models on the complete cases and when you can't. And it comes down to outlining the assumptions that you believe that you have to commit to in your analysis. And there are two types of missing at random assumptions that show up here, one for the censoring, one for the sampling. So that's yeah.
I see. Thank you very much. So, to an extent, essentially, if we were to restrict to the complete cases, we're not looking at the complete population. We'd only look at those individuals who were, for example, well enough to complete the second set of sampling. And with these methods on the conditions, you try to correct that and get back to your original question on the whole recover study.
Yeah, exactly.
Right, great. So then we have a second question. So this is if you know of available MECFS data which could be analyzed using the approach you nicely outlined here to address the specific question of interest. And if you're not aware of that, what data would you need?
Yeah, this is an interesting question. Thank you. I have not really worked much in MECFS as was noted at the beginning of the talk. I've collaborated a little bit with folks here, but on this in this workshop group. So I'm not aware of MECFS data resources that would be amenable to this kind of analysis. But the data that we would need, so that would be some large data resource like the Recover observational study. So this would be a large observational study of individuals who have some kind of MECFS symptomology. You could study the impacts of particular either treatments or other exposures. The A that I have here on the outcome could be particular MECFS outcomes. It doesn't need to be biomarkers. But as long as you have a large data resource where there's some kind of sampling process at play, then these methods become relevant.
Okay, brilliant. Thank you very much, Nima. So I think then we'll move on to our second speaker of the day. So thank you again. So then I'd like to introduce Jiabo Xu from the University of Glasgow. And she is a lecturer in biomedical engineering there, and she leads a research group developing AI-enhanced single-cell technologies for disease diagnostics, combining Raman spectroscopy, microengineering, and machine learning to map immune dysfunction across complex conditions, including MECFS and other autoimmune diseases. So, Jiabo, I'll hand over to you.
Thank you very much, Short, for the nice introduction. And hopefully you can all see my screen. I pointer moving. Okay. Good afternoon, everyone, or good morning. good evening if you're in other parts of the world. My name is Dr. Jiabao Xu. I'm a lecturer in biomedical engineering from the University of Glasgow. And it is my pleasure to speak in this first Prime workshop, which I think is a great initiative, as research should never be isolated, and it's all about communication and participation. So today I want to talk to you about decoding invisible illnesses. We're going to explore how we're using AI and single-cell data science to decode these conditions, from ME/CFS to autoimmune diseases, and move toward precision medicine. So let's start with the scale of the problem. Conditions like ME/CFS, it affects millions of people globally, with an estimated prevalence of 0.1 to 0.8% worldwide. And we are also seeing a massive new wave of post-acute infection syndromes, or PASPAS, triggered by long COVID. But despite the millions suffering, there's a massive diagnostic gap. So patients often look fine, but they are bedbound and daily life hugely affected by this invisible illness. So the most frustrating part for many patients is the normal test result. A doctor runs a standard metabolic panel checking glucose, thyroid, and iron, and everything comes back within the normal range. But then the reality is the chronic pathology, debilitating fatigue, PEM, and autonomic dysfunction. So our goal really is for clinicians and patients and researchers to move away from these subjective surveys and find objective, quantifiable molecular biomarkers. So to find these subjective markers, first we need to understand what connects these conditions. It often starts with an infection. In around 75% of the ME/CFS cases, a viral trigger is reported, and this sets up a chain reaction. Immune dysregulation leads to metabolic remodeling, causing, for example, mitochondrial dysfunction and oxidative stress, and ultimately leading to chronic symptoms. And these changes in immune systems are real. There has been extensive literature which confirms that immune cell dysfunction in ME/CFS as well as long COVID is biologically real. So to find, just to give an example of this literature, such as the 2021 HOEL study, performing comprehensive serum biometabolomics. So in this study, patients did not cluster around a single abnormal pathway. Instead, with 159 metabolites, this patient separated into 3 distinct metabolic states, 2 of which reflected fundamentally different energy strategies. So in the M1, they've called it starvation state. It resembles what we see during like prolonged fasting, i.e. starvation of energy. In contrast, the M2 cohort is in a storage stage, and the patients show a different pattern where the energy substrates like lipids and amino acids, they accumulate, but the cells are failing to access it. And I think the most thing I want to stress here is this kind of heterogeneity among patients explains why a one-size-fits-all diagnostic test has never worked. So the patients aren't all experiencing the same metabolic fault. Now, when we zoom from the blood chemistry down to the single immune cell level, this makes biological sense. Immune cells, especially T cells and NK cells, they're among the most energy-demanding cells in the body. And under healthy conditions, they flexibly switch between glucose metabolism and oxidative phosphorylation depending on the demand. But then in ME/CFS, the mitochondria, the cell's power plants, they lose efficiency. As a result, the cells are forced into a metabolic survival mode. So they switch away from glucose towards burning fats and amino acids, which is slower and less efficient. And this is not a sustainable strategy, and the consequence is immune cell explosion. So when these metabolically stressed cells are pushed, for example, by physical or cognition exertion, they cannot meet the sudden energy demand. And this failure at the cellular level maps directly onto what the patients experience clinically as the PEM post-exertion malaise. So PEM, it is not a failure of motivational conditioning. It is a predictable outcome of bioenergetic collapse at the immune cell level. So it seems like we have increasing amount of knowledge of the immune dysfunction at cellular level. But if the immune cells misbehave, why do standard blood tests miss this? So the blood test should test immune function, right? But if we think a standard blood test like a fruit smoothie, if you have one rotten grape in a giant blender of all fruits, you will never taste the rot. So the signal is averaged out. And this averaging effect is exactly why so many ME/CFS studies contradict each other. Different cohorts, they can have different proportions of these dysfunctional cells, so the bulk signals cancel out. So what we really want is, instead of averaged fruit smoothie, is to look at a fruit bowl. So where we can pick up every individual piece of the fruit and identify the bad ones, even if they only make up, for example, 5% of the population. And this is exactly why we use single cell analysis, which I will show very shortly. So in conclusion, we must zoom in, improve our resolution, and interrogate cells one by one to find the metabolic faults. To do this, we use a technique called single-cell Raman spectroscopy. It is label-free, meaning we don't need expensive dyes or antibodies. We simply shine a green light on the cell, and the way that light scatters, called inelastic scattering, provides a unique molecular fingerprint of everything inside the cell, its protein, lipids, and nucleic acids. And this is what a cell's fingerprint looks like. Every peak represents a specific chemical bond. For example, the CH region is dominated by lipids, and by looking at this region, we can observe the lipid shift, which can be crucial to observe the burning lipids instead of glucose behavior that occurs in ME/CFS. Now, with the Raman fingerprint, this creates a massive amount of data, over 1,000 data points per cell and hundreds of cells per patient. So to make sense of it, we use an AI engine to detect metabolic patterns that are far too subtle for the human eye to see, and then build machine learning classification models for diagnosis. So here, maybe I will briefly stress something important about the AI practice here, because this is our theme today. So for AI practice, we should always use strict patient-level separation, like in here. This is especially relevant for single-cell analysis, because if we're doing a cell-level split, and the model was trained using some cells from a person and tested using the rest of the cells from the same person, this will be cheating, as the model has already seen that person. We should always have how-to test set to avoid data leakage. And in addition to that, our team also prioritizes interpretal features, not just accuracy. So Raman is not a black box technique. Every peak corresponds to a known chemical bond. So when a model highlights a region, we can map that directly back to, for example, lipid metabolism or amino acid usage. And this is important because for clinical adoption, understanding why a model makes a decision is just as important as the accuracy, the performance. So this leads to the first case study I will talk about today. In 2023, Becky was a researcher at Oxford. We published a study in Advanced Science showing a high accuracy diagnostic test for ME/CFS using patients' blood immune cells, so PBMCs, and their single cell Raman fingerprints. For the study design, we compared 61 ME/CFS patients against 16 healthy controls and 21 patients with multiple sclerosis, MS. So we included MS patients specifically to prove that the AI wasn't just detecting general fatigue, but rather the specific metabolic hallmark of ME/CFS. So we isolated the PBMCs, which are the immune cells from individuals' blood, collected a total of over 14,000 spectrum, and built a ensemble learning AI model for diagnosis. And the results were remarkable. We achieved a 91% sensitivity and 93% specificity for diagnosing ME/CFS, comparing with healthy controls and disease controls MS. Furthermore, our model was 84% accurate in distinguishing between mild, moderate, and severe cases of ME/CFS, so differentiating between disease severity. And because Raman fingerprints measures chemistry, we can see why the patient's immune cells are sick, i.e. the explainable AI I mentioned. We found elevated tryptophan, suggesting a metabolic trap, where cells cannot process energy correctly, like how the other studies reported. We also saw reduced glycogen and elevated glycerol, providing these cells are energy stuffed and are literally breaking down their own fats to survive. So this study hopefully started one of the first steps that moves us from a world of current standard of stigma and doubt, where the patients are told it's all in your head, to a world of objective biomarkers and actionable metabolic targets. And for patients, the shift from subjective complaints to objective biology is not abstract. It determines whether someone is believed, supported, and included in research. So moving on from the ME/CFS study, it inspired us to step into more immune-related diseases and enabling precision medicine from a single cell point of view in these complex diseases. So the second case study I will cover today is an ongoing study. In this study, we applied the same logic to systemic lupus, so SLE. SLE is an autoimmune disease that is highly unpredictable and heterogeneous. So it can attack the brain, heart, kidneys, or skin. So here clinically, we can see we have at least 5 different clinical subtypes with symptoms attacking different parts of the human body. And current blood markers lack specificity for predicting which organ will be attacked next, and confirming organ damage, for example, damage to the liver, currently requires invasive tissue biopsies. And these cannot be performed frequently for monitoring, and this leaves clinicians reactive rather than proactive. So because the results haven't been published and we're still collecting a separate large validation cohort, I will just give a high-level overview of the results. So similarly, as in the ME/CFS study, we used SLE's patients' PBMC immune cells and their Raman fingerprints. We have trained a specialized deep learning model called PBMC-NET. This model successfully stratified patients into 5 distinct clinical subtypes, with around 80% accuracy at the cell level, which is the top figure here, and remarkably 100% accuracy at the patient level in the bottom figure. This means that we can predict which organ might be at risk before the damage becomes irreversible. Finally, for the last case, we look at psoriatic arthritis, PSA. So in PSA, doctors will give treatment to treat joint inflammation and pain. However, around larger than 50% of the patients, even when their inflammation is treated, their fatigue persists as an invisible symptom. So imagine this debilitating fatigue happens in half of the clinical remissions. So the question here is fatigue simply a secondary symptom of pain, or is it a biologically distinct process? To answer that, we performed a correlation analysis of Raman spectral features against clinical scores for pain, for inflammation, or for fatigue. And this analysis found that there are effectively zero overlaps between the pain spectrum and the fatigue spectrum, meaning that the chemical vibrations of fatigue are different from the chemical vibrations of inflammation. So they are entirely separate pathologies. So biologically, fatigue isn't equal to inflammation. They are separate pathologies co-occurring in the same patients. And because we're looking this at the single cell level, we also trace the source of this divergence. So inflammation is driven by CD8 plus T cells, which currently drugs target wall, but the fatigue is driven by CD4 plus T cells, a different subtype of immune cells, and they are suffering from oxidative stress. And this is a pathway currently the anti-inflammatory drugs do not touch, which explains the unresolved fatigue after treatment, even when the inflammation is resolved. So these two studies on SLE and PSA, followed by our ME/CFS publication, although still ongoing, we're moving forward from just diagnosis to solving clinical subtypes, as well as decoupling symptoms of pain and fatigue, and finding immune cell subpopulations responsible for the two different pathways. So I think a message I hope to convey is each individual is different. This is true for each patient and even each immune cell, like the fruit smoothie and fruit bowl analogy I mentioned at the beginning. So for these chronic invisible illnesses like ME/CFS, like long COVID, and many more, we should really be moving from managing general symptoms to targeting specific cell type mechanisms. So moving from one treatment fits all to true precision medicine targeting individuals. And this final page hopefully demonstrates our vision to turn our methodology into a universal platform. Imagine a single blood sample being run through a modular AI model, AI platform, and one AI software module checks for ME/CFS, another for sepsis, and another for autoimmune risk. And what is important here is that it is not a single diagnostic test. The infrastructure, single cell phenotyping using Raman fingerprints plus modular AI stays the same. What changes is the disease-specific training. This means that this platform can continuously improve as more data are added, and hopefully this will become a scalable, precise, comprehensive way to monitor immune health. So these studies are collaboration between the University of Glasgow, Oxford, and Peking University People's Hospital. And I want to thank the patients and my co-authors for making these work possible. And thank you very much for listening.
Thank you very much for that interesting talk. That's really great. So we have a number of questions in the chat. Let me start off by one question regarding case study 1, where you mentioned that there was a 20% independent set of held out data. And the question is, was that 20% independent set indeed never used in training the model? So was it truly held out?
This is a great question because for a small sample cohort size like our study, we actually use cross-validation. So we use 5-fold cross-validation, meaning that we split the cohort into 5 different folds, and we use the 4 folds to train the model, and the 1 fold the patients never seen by the model during the training process to test the model. And then we repeat this process using the other 4 as training and a separate one for testing. So we do it for 5 times so that every patient actually is being tested, but then during the training process, it's never entered the model training to avoid the data leakage. Hopefully this is clear of our approach.
Yeah, great. Thank you. And I think there's a follow-up question in this regard, which is for case study 2, with the remarkable results of 100% accuracy on the lupus patient stratification. So the person asking this question says that these results give them some pause. Usually, when there's 100% accuracy, this is either due to indicative of overfitting, so perhaps it's unseen data still used in the training. And the second part is what has been done to check that patients are not stratifying based on other features rather than the labels you use, such as sex, age, or other comorbidities.
This is a very good question again. Yes, 100% is always make people doubt. But our approach is definitely patient level. First of all, definitely patient level split. So the single cells from one patient will just belong to that patient and will not enter both the training and test sets. And also we split the patients into training and test so that one patient never enter both of the sets. But again, this is the first data set we have from one hospital, so one cohort only, and we are now collecting a validation data set from 2 other hospitals, and hopefully this 100% will, even if it decrease, it will all still stay robust with the other cohorts from other hospitals. And for the second part of the question, the confounding effect is always a problem. I didn't show it here, but we ran already for sex, inflammation parameters, other clinical assays as confounding effect, and we will make sure to fully report these during the publication process.
Nice, thank you. And have you compared the results from this model to sort of baseline statistical models, say a linear regression with regularization as usually recommended to ensure that the complexity of the model is actually relevant and used?
Definitely, we use traditional machine learning as the baseline to compare our deep learning with. For example, for Raman single cell data, SVM, support vector machine, is usually very robust against overfitting in smaller data sets. So we compared our deep learning with the support vector machine algorithm, and we show better performance.
Right, great, thank you. So someone is also asking if this Raman model is open source.
Yes, we for the only paper we published already is the first case study, and the code data sets are all open access. And for the other 2, we will definitely make sure it's open access to everyone.
Great, thank you. Yeah, so and finally, so I had a question regarding the Raman fingerprint. So could you say something about how unique this fingerprint is? By which I mean if you have 2 different cells that are biologically distinct, how do you know for sure that their Raman fingerprints are not indistinguishable?
I think the question maybe is it is too sensitive as in distinguishing different cells. For example, some differences that we might not want to detect. For example, when you have an infection, you have just an inflammation, the immune cells will have different response, and the Raman detects that. And we want to make sure that it is detecting the disease-specific fingerprints instead of just the inflammation, right? So I think the sensitivity here is maybe just too high and makes the fingerprint noisy. So what Raman detects is the molecular vibrations inside the cell. So it collects all molecules and their molecular vibration, chemical vibrations, and reflect them all in one fingerprint. So sometimes it can be over noisy and too many features, and we get lost in the sea of information. So I think this is where the AI, the machine learning or deep learning can really help to extract those meaningful differences in this noisy kind of too sensitive fingerprint.
Thank you. And there's a further question. So why is Raman a preferred technique for this rather than other single cell analysis techniques such as mass spectrometry?
This is a very good question. So we actually use metabolomics, transcriptomics, so sequencing technique, omics technique in our study as well to provide a comprehensive kind of view. And sometimes adding this will improve the biological interpretation, of course. But then Raman is especially preferable as a diagnostic test approach because it is label-free, it is relatively inexpensive, and so in all our studies, we use just the peripheral blood. So it is a minimally invasive and relatively cheap, relatively non-expensive technique. And I think this has advantages for a diagnostic test.
Great, thank you. Excellent. There's another question. So regarding case study 1, this is a follow-up question. So Chris is saying, I would be totally convinced of its accuracy if your model had never seen a truly held out data set that was used for testing. Is there any intention to do this soon? If not, what would you need? Because Prime might know someone to contact.
That would be great if we can have more validation or test sets actually to improve our further convince people to turn this as a real diagnostic test. And I think, like I said in the LSE data, although during model training and testing we have a separated test set. This was from the same cohort, for example, patients from the same hospital, or the patients from in the MECFS study, we used the UK Biobank data, right? So it would be nice to have another sample, another cohort, for example, from another country, from another database to further validate our study. So it'd be great if Chris can help us connect to resources like this. That would be very much appreciated.
Excellent. Thank you very much again for your talk and for answering these questions. This was very helpful. Great. So that means we move on to our third talk of today's workshop, which will be presented by Philippe Boileau, who is an assistant professor of biostatistics at McGill University and who is joining us somewhat in the morning from his perspective. So he's interested in the development of assumption lean statistical methods and their application to quantitative problems in the health and life sciences. And he has recently focused on creating such methods for heterogeneous treatment effect discovery in clinical trial data, which I believe is what he's going to be telling us about today. So Philippe, the floor is yours.
Yeah, thank you so much for the introduction and the invitation to discuss some of my recent work. So indeed, we'll be talking about applying these methods to clinical trials today that are hopefully relevant to some of the questions that many of you are asking relating to ME and CFS. All right, so this work was done in collaboration with Hani Zaki and Marie Schnitzer at the University of Montreal and Tanya Janotis Ferreira at the Research Institute of the McGill University Health Center, also in Montreal. And so the motivation for this methodology and this talk really stems from my experience working in pharma before I came to academia. And what I've done basically is I encountered a number of I think, very interesting methodological problems that I just didn't have time to really dig into while I was working in private industry. But now that I'm working in academia, I do have time to sink my teeth into them. And so in particular, what I'm interested in is understanding how heterogeneous subgroups drive heterogeneous treatment effects, particularly in clinical trials, as was mentioned. All right, and so to motivate this entire methodology, I'm going to work from the environment of a drug development pipeline, but just note that this is broadly applicable to other settings as well. All right, so we're going to start off with a simple sketch of a phase 2 clinical trial where there are two arms, a treatment arm with a novel therapy and a control arm with the standard of care. Patients are randomized with equal probability to each arm. The people in the treatment arm are denoted in blue in the scatter plot, and in red if they're in the control arm. And these patients are plotted against their baseline covariates, all right? In particular, two baseline covariates. And these could be something as simple as age or some more interesting biomarker related to, for example, their omics profile, all right? Now, importantly, in this sketch, we're going to assume that these subgroups are either unknown or unaccounted for, meaning that the baseline covariates are either not measured or they're measured but we're not using it in the design to somehow account for the subgroups that we're seeing on the screen. Now, it's not unreasonable to think that these subgroups represent different phenotypes of patients and that these different phenotypes will actually respond differently to whether or not they receive the medication in the treatment arm or the medication in the control arm. And so what I mean by this is, if you can see my pointer, is that the patients in this top left cluster tend to benefit more on average from receiving the treatment than the control. And the same is true for this, this cluster of patients or this phenotype in the bottom left corner. On the far right in the bottom, however, we see that we have this group of patients, it's relatively small, that really does not benefit from receiving the novel treatment compared to the standard of care. Now, because we're not taking into account the baseline covariates, we're also not taking into account these subgroups and their individual treatment effects. Instead, what we're going to do at the end of this trial, once we're finished running it, is take the weighted average of patients' response, essentially, and end up with this marginal effect, which shows that on average, in this patient population or in this indication, that patients will respond a little bit better to this novel treatment as compared to the standard of care. All right. So in the drug development pipeline, this is a success. This means that we can move on to a phase 3 trial. So a slightly larger trial where now we're trying to really nail down comparative efficacy. All right, which will eventually dictate treatment guidelines and the release of this new drug on the market. Now, in this simple sketch, we're going to assume that we basically are random sampling from a very similar population as the phase 2 clinical trial, but not identical. And I mean not identical because although we have this same exact patient phenotypes in this new phase 3 trial, this previously small subgroup that responded negatively to the new to this new novel trial medicine is now more represented. And so again, we see that these subgroups have the exact same treatment effects as we saw previously. So we're going to assume here also for simplicity that the endpoints across these two trials are approximately the same, or at least that the phase 2 endpoint that was used was a good surrogate for the endpoint used in this comparative analysis. All right. But again, we're not taking into account these baseline covariates, these baseline biomarkers. And so we're only going to take the or compute or estimate the marginal effect within this trial. Now, because of the change in weights across these subgroups, we're now going to have a slightly different story. We're now, instead of the new trial medication being better on average for patients than the control, we see the opposite. All right, we see that on average, patients actually respond better to the control than the novel treatment. And this would represent a null trial or a failure in that you wouldn't be able to move forward and necessarily recommend to patients or change the guidelines that say that everybody should be receiving the novel treatment. All right. So this is obviously a bad outcome for investigators, for trial sponsors, but also for patients. All right. Because that just means that this new medication that we know actually does work for some people is not going to be available to the people that it actually benefits. So what's going on here? Why are we seeing this heterogeneity? Well, simply put, our baseline covariate 1 here is a treatment effect modifier. And that means that it's a baseline covariate that interacts with treatment assignment, and that this will actually dictate how people will respond to treatment on average. All right. And so you could actually plot out patients' predicted outcome as a function of baseline covariates and what arm patients are assigned to. So if you have a small value of baseline covariate 1, then you're going to end up in this region of this simplified plot. And we'll state that on average, patients with this profile would actually benefit more from receiving the new therapy than the standard of care. If you have a large value of baseline covariate 1, then it's the opposite interpretation. Now, if we actually had access to this knowledge that baseline covariate 1 was an effect modifier, then we can modify our trial design to only enrich for patients who actually have smaller values of baseline covariate 1, and then downstream also modify guidelines to make sure that patients who do have larger value of baseline covariate 1s receive the standard of care and not this new treatment. Now, again, this is in the context of like a drug development pipeline, but this applies to all kinds of different settings, including, for example, potentially running a successful phase 3 trial where you identify a meaningful treatment effect. But then when you transport this treatment effect or this intervention to a more heterogeneous population in the real world, you might have some change in the average treatment effect, and you'll want to account for that change by accounting for the treatment effect modifiers. And then again, modifying guidelines and treatment procedures based off of your knowledge about these effect modifiers. So overall, it's just really important to have some background knowledge about these kinds of baseline covariates. But the issue is that it's very challenging to get a handle on them, all right? And so standard procedures exist, and they work well, but only when you actually have access to the true effect modifiers in your data set, all right? But there's no guarantee that you actually have these effect modifiers in your data set, because sometimes, as we saw in previous talks, these effect modifiers are just not known a priori. We have to understand the mechanism of the disease, the mechanism of the treatment, to actually want to collect them. Sometimes we do know what these effect modifiers are, but due to resource constraints, we might not actually prioritize collecting them. And sometimes even if we do have the resources and we have the knowledge, we might not be able to collect them properly due to mismeasurement error or technological limitations, all right? And so the question we're going to try and answer with the talk and with the methods that we developed that we'll be presenting is, can we still uncover evidence of a heterogeneous treatment effect, even if we don't have access to treatment effect modifiers, all right? And obviously, if I'm talking to you about this today, the answer is yes, and then I'll kind of give you the details, all right? So we'll go over the problem formulation, and I'm not going to try and bog you down with too much math, but we'll just assume that we have access to this, for now, idealized data set, all right? Or we have N observations or N patients in our clinical trial. We've collected P baseline covariates, which we've labeled W1 to WP, and these could be categorical variables that are transformed into dummy variables. That could be binary variables like W1. They can be continuous like WP. There's really no limitation here, all right? We're then also going to assume that we have a binary treatment indicator representing random assignment to a treatment arm or a control arm. And finally, we'll assume that we have access to potential outcomes. Now, Nima did a great job of introducing what these potential outcomes are, but just to repeat, these are basically the outcomes that you would observe for each different observation under alternative treatment assignment rules. So for example, this first observation over here, had they received not the novel treatment, but were instead randomized to the control arm, then their outcome would be 2.5. If they were randomized to the treatment arm, then we would have a value of 5.1. And we get to observe these for every single individual patient, and that's what we call this individualized data set. And importantly, for the context of this problem, we're going to assume that these outcomes are continuous, all right? They can be bounded, but they're continuous. Now, of course, it's idealized because in reality, we're only going to get to observe the potential outcome associated with the correct treatment assignment. So for patient 1, you would only see Y1. For patient 2, you'd only see Y0. Same for Y3, for observation 3, et cetera, et cetera. All right? But if you bear with me for a moment, I'll explain why this is important. All right? Now, if we actually had access to these potential outcomes, what we can do is compute for each individual patient their individual treatment effect, which is nothing more than the difference of the potential outcome under treatment minus the potential outcome under control. All right. And this gives the benefit for every individual from receiving treatment over the standard of care. All right. Now, of course, if we had many, many observations, say N was large, or even had like a population level data set, then this column in this idealized spreadsheet would not really be all that useful. We need to come up with a way to summarize it. All right. And so the standard way of doing this is to use a causal parameter known as the average treatment effect. Again, Nima did a great job of introducing this, but the main idea here is that it just tells you what is the average outcome under treatment minus the average outcome under control if all individuals had received treatment versus if all individuals had received control. All right. So it's a nice summary metric of a treatment effect. But it's just a summary of the treatment effect. It doesn't actually get at what is driving heterogeneity. What is the mechanism of heterogeneity? To get at that, we have to use something called the conditional average treatment effect, all right? Or at least we can use it, and that's a standard practice in the causal inference literature. And so the conditional average treatment effect is really nothing more than a regression of this ITE column, the individual treatment effect column, on the W's that we have in our data set, all right? And so if we want to represent this mathematically, it's simply the expectation of the difference of the potential outcomes conditional on W, all right? Now, again, this is arguably one of the most popular parameters in causal inference to uncover effect heterogeneity. And the reason for this is that you actually get to understand the mechanism of this heterogeneity if you're able to estimate the K properly, all right? But that's a big if, because again, this is a conditional parameter on W. So to actually uncover heterogeneity with the Kate, you have to assume that there is true effect modifiers in your set of W's that you have access to, all right? Which again is not always the case due to a myriad reasons. You might think, okay, well, if I really have to make sure that my W's contain a true effect modifier, maybe my best bet is just to throw the kitchen sink at Kate estimation and consider an enormous number of W's, right? Unfortunately, that's not an amazing approach either, unless you're willing to make some very stringent assumptions that simplify the estimation procedure, such as sparsity, where there's only a few W's out of the potentially thousands that you're considering that are truly driving effect modification, all right? And if those assumptions are not satisfied, you'll end up in a situation where you actually have a lot of false positives, all right? And so we want to come up with another causal parameter that allows us to tackle this question or identify heterogeneity without relying on the measurement of W's, all right? So in this sketch so far, I've been taking for granted that heterogeneity is present, all right? But let's take a step back and consider what it means to have a homogeneous treatment effect, and this will kind of set up our solution to this problem, all right? So in a homogeneous treatment effect setting, what we're effectively saying is that the ITE is identical for all individuals, all right? And another way of putting this is that the potential outcome under treatment is nothing more than the potential outcome under control for all individuals plus some shift of delta. All right. And in this case, the delta is equal to the ATE. Okay. So now that we've defined what a homogeneous treatment effect setting is, can we use this hypothesis or an assumption of a homogeneous treatment effect to determine whether or not it's reasonable or if there's actual evidence for effect heterogeneity? All right. And so the answer is yes, we could actually construct formal hypothesis testing procedures for this. We have some nice math to prove it. I'm not going to show you that math. I'll just provide some visual interpretation, all right? So starting in this top left-hand plot over here, we're going to work in this homogeneous individual treatment effect setting, all right? And that means that every individual in our population has the same ITE. And this ITE is centered around the average treatment effect. So really, it's a distribution of ITs, but just focus at a point, all right? And so if we want to actually plot what the potential outcome distributions look like for this setting, what you would see is something that looks like this, all right? Where you've effectively shifted your potential outcomes under control by some gamma to the potential outcomes under treatment, all right? And so for any individual in our data set, we know that if they have a potential outcome under control at this red line, then the potential outcome under treatment is equal to this blue line. And the same is true for all individuals in the data set without question, all right? And you'll notice that from this plot that the distributions of our potential outcomes have this nice property of identical variances, identical spread, all right? If we have a potentially heterogeneous treatment effect setting, or at least we're not willing to necessarily assume a homogeneous treatment effect setting, then we could also plot the distributions of these individual treatment effects, of these potential outcomes, and we might note that the variances are different, all right? And if we see that the variances are different, then we might try and do the same exercise of trying to figure out, well, how do we visually shift patients from the potential outcome under controls to the potential outcome under treatment? And the reality is there's no obvious way of doing this, all right? We don't know if this patient represented by the red line is a shift to the right or a shift to the left. And indeed, what you can do is actually compute the ITEs from these potential outcome distributions and see that you actually have some spread in the ITE distribution. And because you have some spread, you know that you have heterogeneity, all right? Not all patients extract the same benefit from the intervention, And so the bottom line here is that if you have potential outcomes with different variances, you must have a heterogeneous treatment effect. All right. And so what we've done is we've encoded this information in some new causal estimates, some new ways of summarizing that we have heterogeneous treatment effects. And we'll call these differential variance estimates. All right. And there are many ways to define these kinds of estimates. We've considered two. The first is the absolute differential variance, which is a bit of a misnomer because it's really the difference of standard deviations of the potential outcomes. And then we have a relative differential variance estimand, which is the ratio of the potential outcome variances. And the reason this is interesting is because from these estimands, we can construct formal hypothesis tests about homogeneity. All right. So if we're using our absolute differential variance estimand, if the treatment effect truly is homogeneous, then we know that this psi c is equal to 0 because the standard deviations are equal to each other, or equivalently, the variances are equal to each other. But if we have evidence that this is not the case, that psi c is actually not equal to 0, then we're in the presence of a heterogeneous treatment effect. And the same can be said for our lambda, all right? Except instead of zeros, we're going to have ones. Now, of course, the elephant in the room here is that we don't actually ever have access to these potential outcomes, right? So this is still an idealized setting. It's not clear that we can actually get access to these causal estimates, right? In reality, we only have access to this column Y. Our potential outcomes are unavailable. But it turns out through standard identifiability conditions, which again, Nima introduced in his presentation, it turns out we can still estimate or infer these causal estimates from observational data without our potential outcomes, all right? I'm not going to provide the details here simply because I'm running out of time, but just note that we can estimate them in clinical trials, all right? And these assumptions are guaranteed to be satisfied so long as we have randomization. So now how do we go about estimating these estimands from our observed clinical trial data? Well, we've decided to go with causal machine learning estimators, though it's definitely not the only approach that we can use, all right? And so in this work, we derived targeted maximum likelihood estimators, one-step estimators, which are equivalent to double D-biased machine learning estimators in this setting, and then we've developed cross-fitted equivalents of these estimators as well, all right? Now, these are called causal machine learning estimators because they can rely on machine learning estimators to estimate some of the machinery that's hidden under the hood, the nuisance parameters need to be estimated. And by using these machine learning estimators, we get away from making any parametric assumptions about how the data is being generated, while still maintaining the ability to get formal inference in statistical terms. So for example, controlling type 1 error rate when we perform our hypothesis tests. And so when we perform inference with these estimators, even though we're using flexible machine learning models, we still have rigorous, robust procedure for making data-driven decisions. And we understand how they behave asymptotically. And so we can control our false positive rate. I'm not going to go over simulations. I think I'm running out of time. So I'll just quickly talk about the application section of our paper on this. And so Tanya, one of the collaborators on this project, ran a clinical trial in long COVID trying to assess whether virtual physical rehabilitation for patients living with long COVID is actually something that can improve their mobility scores. All right. So this was an 8-week virtual home-based physical rehabilitation program. And the goal again was really to see if we could improve functional mobility in adults with long COVID compared to the standard of care. All right. And so 132 participants were enrolled across 2 Canadian sites. And the intervention was doled out in the treatment arm. This intervention was supervised by physical therapist professionals. All right. And so unfortunately, the final trial readout was null, meaning that this intervention did not show evidence of any improvement in an AMPAC score, which again is just a measure of functional mobility, though there was some signal that this intervention could improve health status and symptoms of specific measures. All right. And so the question that Tanya had was whether or not there might actually be pockets of patients or subgroups of patients that truly do benefit from this virtual rehabilitation program, given how heterogeneous this patient population is. And so we started off by analyzing the data with traditional methods Kate estimators to try and uncover the mechanism of heterogeneity if it was truly present. And unfortunately, those results were also null, no matter what methods we threw at the problem. And so as a sensitivity analysis, we decided to apply our proposed approach of differential variance inference. And so in particular, we focused on trying to estimate or infer the absolute differential variance using our targeted maximum likelihood estimator. All right. And so that's given, again, just to remind you, by this target parameter over here, the difference of the potential outcome standard deviations. Now, our estimate of this parameter was 3.38, which, on its own suggests that there's a slight inflation in terms of the variance of the outcomes in the treatment arm of the patients who received this novel intervention compared to the standard of care. But then when we perform statistical inference by getting our 95% CIs and computing p-values, we saw that, in fact, there was no evidence to suggest there truly was any meaningful difference between the spread across these two potential outcomes distributions. And so here the conclusion is that there is insufficient evidence to reject the hypothesis that this physical rehabilitation intervention had a heterogeneous impact on patients' AMPAC scores. And so with that, I'll say thank you for listening. This method is available for use through a open source R package available on GitHub called SAMLDiffR. It's in its early stages. There still needs to be some touch-ups, but I'd be very appreciative if you can, take it for a spin. Let me know if you think there's any additions that would be made to improve its quality, the user quality experience. And then we also have our paper describing in more detail the actual underlying methodology on arXiv, which is linked on my website. So thank you all for listening.
Great. Thank you much, Philip, for a very, very insightful talk. Yeah, so let me just get the main question out of the way. Do you think this would be a method that can be meaningfully applied to find heterogeneity in individuals with ME/CFS and or long COVID? And what kind of data do you think would be suitable to do that?
Yeah, I think it's definitely interesting. Here, like the main caveat is that the outcome of interest, your primary endpoint, has to be continuous. But if that fits the bill, then you're able to do this approach. And I think it's particularly interesting in settings where the initial study results might have been null, but you believe there might still be heterogeneity. And so instead of going on a fishing expedition trying to identify what the mechanism is for this heterogeneity, you could start your process of trying to uncover these subgroups by applying this method to see if there's any actual evidence of a heterogeneous effect. And if there is, then you can dive into the difficult task of trying to actually understand what makes up these subgroups. And so as I was saying, I'm considering some, in this talk, some abstract biomarkers, but I think this is actually a particularly interesting approach for any kind of data set where you have omics information on patients. So for example, gene expression, epigenetics, you name it. This is a way of seeing if you can detect heterogeneous treatment effects that might be driven by these data sources.
I see. Okay, so something like biomarker data might be helpful as a treatment response here, potentially. Okay, great. Thank you. So there is a question in the chat from Sam. So given the long-standing evidence from ME/CFS research to date, they're curious what a hypothesis calculation might look like when considering the possibility of worsening, I guess, health outcome and not just positive outcomes versus lack of such.
Yeah, so that's a great comment. I'm not an expert in ME/CFS, but I have spoken about this with my collaborator Tanya, and she had pushback when she was proposing this trial and running it. And I think it's important to note here that this physical rehabilitation program is really catered to the patient to make sure that they avoid these negative outcomes. So patients who might exhibit post-exercisational malaise would actually have a reduced burden of physical physical rehabilitation through this program to try and avoid this issue. But with that said, that's also a potential driver of heterogeneity. We just weren't able to uncover any evidence of that actual heterogeneity from this clinical trial data set. And that's not saying that it doesn't exist. It's just that in this data, given our admittedly limited sample size, we couldn't detect it.
All right, thank you. Yeah, and Chris is adding to Sam's comment and question that MECFS research shows clearly that exercise tasks like this are commonly harmful, that symptoms are exacerbated. So those long COVID people with post-exertional malaise, for example, would be particularly negatively affected, but those without it might have positive outcomes. So I guess this could be a driver of heterogeneity. And Nuno mentions that related to the clinical trial, a possible treatment modifier could be individuals complying or not with a MECFS case definition, because MECFS and/or long COVID patients cannot tolerate exercise. Yeah, so that's on a similar note. Great. So one other question I wanted to ask. So if I understand correctly, when there is a null effect on average in a study, this approach can be used to detect if there is nonetheless heterogeneity present. Have you compared this approach to well, so I guess the intent there is somewhat different, but to the recent work of Edward Kennedy and collaborators on clustering, so causal clustering using k-means, where they essentially look at treatment response and try to cluster individuals based on their covariates, age, sex, health status, into clusters that similarly respond to treatment. So I think your initial figure reminded me of that, with the distinction that they would probably separate off these three groups into three distinct clusters.
That's a phenomenal question, and the answer is, this is an approach that's been around for a while. Maybe not formally as well as nicely studied by Ed's group as others, but it has been used for a while. But it still comes down to estimating a conditional average treatment effect, where now the W's you're conditioning on are not the baseline covariates, but some summary of those baseline covariates that you obtain from clustering. And so the bottom line is that if you don't actually have treatment effect modifiers in your data set, then you won't actually be able to create clusters of patients that correspond to heterogeneous subgroups. And so again, like you wouldn't be able to uncover effect heterogeneity with these standard approaches. You'd have to use something slightly different, like what we've proposed here. But that it is super interesting work, and I think very, very applicable to many settings, especially in clinical trials.
Okay, great. Thank you. I think we'll be in touch with you on that point. That would be great to talk about. Yeah, so finally, there's one more question by Nuno before we head into our break. I guess this partially relates to the null result you showed at the end. So is there a sample size calculator based on the method? So I presume this is a required sample size in order to detect heterogeneity, provided sufficient heterogeneity is indeed present in the data.
Great question. It's something that we're working on right now with some master students. So I'll let you know when it comes out.
Okay, great. So excellent. Okay, thank you very much. Certainly all the speakers for now, and also those who asked questions and commented. So we'll be back in about 10 minutes. We're exactly on schedule. So 10 to 4 p.m. here in the UK. And then I'll hand over chairing to Ava Hamsen. So see you all in 10 minutes. Bye.
Hello, welcome back from the break, everyone. So the next speaker is Maria. So she's a cross-disciplinary fellow at the University of Edinburgh, and she works on development and application of statistical estimation prediction models, especially with a focus on uncertainty quantification. And today she's going to talk to us about her work on conformal prediction in the context of ME biomarkers. Thank you, Maria.
Hello. Hi, everyone. Hope you can all see my screen.
Yes, thank you.
Thanks for organizing the webinar and thanks for inviting me. I'm now going to talk about conformal prediction and links to subgroup stratification in ME/CFS blood biomarkers. So I think before I go on, I should be clear about what the talk is about and what the scope is. So basically, today we'll talk about assessing the predictive signal of blood biomarkers for ME/CFS. What I'll show today is a validated way to attach reliable uncertainty to biomarker-based predictions and how that uncertainty looks at the level of individual participants. And I'll also show early evidence that uncertainty aligns with feature attribution. What this talk is not about is not a diagnostic test, is not a claim about definitive ME/CFS subtypes, and the biological interpretation is ongoing. So I'll be explicit about where the results end and where the hypotheses begin. So this work starts from the premise that uncertainty here isn't optional, it's essential, and most of you will recognize the challenges. ME/CFS is highly heterogeneous, both within and between patients. There is no signal definitive biomarker, and there is a substantial overlap of blood biomarkers with related conditions. On top of that, the data sets that exist tend to be small and noisy, and this combination is risky from the modeling perspective because it makes very easy to build models that look confident, but they're actually not reliable. So we'll discuss it in a few slides, as you see. So all the analysis I'll present today are based on the UK Biobank data. The aim of this work, as I mentioned, is to evaluate whether measured blood biomarkers contain predictive information for ME/CFS at the population scale. We use two complementary feature sets. First, standard clinical blood traits such as C-reactive protein, cholesterol, or blood cell counts. And second, NMR-based metabolic measurements, which include detailed lipid fractions, fatty acid composition, and lipoprotein particles concentrations, which basically capture broader metabolic processes. Case control definition rely on multiple strands of evidence, including self-reported ME/CFS, non-discordant responses to prior ME/CFS diagnosis in the pain questionnaire, and overall self-rated health, as you can see here. So what most of you will be familiar with is with traditional machine learning, or ML, as you will see along the talk, which most models produce a single label for each individual, for instance, ME/CFS or non-ME/CFS. So basically, that forces the model to make a decision, even when the data don't really support one. And instead, what we can ask is a different question, which is which labels are statistically plausible at a specified uncertainty level. So here what you see is how conformal prediction answers that, which returns a prediction set. The prediction set can contain only ME/CFS or non-ME/CFS, but for others it contains both, which is a principal way of saying we don't know, which would basically happen also to a doctor or a healthcare practitioner. Importantly, this uncertainty is part of the input and it's not something we infer after the fact. So before introducing conformal prediction, I want to briefly clarify best practices for model construction and evaluation. In standard prediction, model fitting, feature selection, hyperparameter tuning, It's basically using, well, must be actually confined to the training data. And it's usually done using cross-validation. And then the final performance metrics, which is normally what we say in the papers or when a model goes public. So as we can say, AUC, AUCPR, accuracy, it must be reported on the holdout set. In ME/CFS and long COVID prediction studies, we often see cross-validation applied in ways where the data used for final performance evaluation is not fully disjoint for training, and that leads to over-optimistic results. An easy way that I like to explain that is basically the way you train a model could be a little bit on how children learn. You show them a pattern of blood biomarkers or NMR, and you tell them those belong to someone with ME/CFS. And then you show others, and you tell those belong to someone with non-ME/CFS. And you show the model lots of patterns, and you try the model. So the model tries to infer information from it. What happens if we show to the model data that we will later be using to know how good the model is, the model will have had the chance to memorize those patterns already, so it will always be better than what it actually would be with someone they would have never seen. So conformal predictions builds on this setup. And here we can see the logic of conformal prediction. We first train the model using a separate training data. Then we apply this model to a separate calibration set to calculate the non-conformity scores or the calibration score distribution. That will give us a distribution that then we can threshold. And when we test individuals which are outside of any of those sets, we can get them the different prediction set labels that I mentioned. Those labels will be, well, those labels that appear will be taken from the distribution that we saw before. So they fall under the threshold. So the result is a prediction set with a formal error guarantee. In addition to careful data splitting, I also want to be explicit about the models we use. Within the training set, we apply a superlearner ensemble, which means that we not only train our data, we not only train one model with our data, but we train several complementary algorithms, including linear models such as Ridge, Lasso, and Elastic Nest, as well as nonlinear models like Random Forest and XGBoost. Importantly, all hyperparameter tuning and regularization and any feature screening happen inside the inner cross-validation loop, and none of these steps ever see the calibration or the outer test set. This step avoids double dipping and ensures valid performance estimates. And it's particularly important when we later want to attach uncertainty to individual predictions, as we are doing with conformal predictions. So as you may have seen in the previous slides, it appears alpha. This is the parameter that controls how conservative the prediction sets are. Formally, it's the missed coverage rate, which is the fraction of individuals for whom the prediction set is allowed to miss the true label. So if alpha is 0.1, what we are expecting is that the prediction set contains the correct level for this participant at least for 90% of the time. So choosing a smaller alpha, it makes the model more conscious. It makes the model more conscious. For example, cautious. For example, we can ask the model to be right 99% of the time. So that means that it's very likely that the model tends to add the two labels to the label set because it's not fully sure at this level of certainty what is the classification of the patient they're seeing. And then we can also go for larger alphas, but that will produce smaller prediction sets, and it will be at the cost of tolerating more errors. Importantly, alpha is not learned by the model. It's a user choice, and it reflects how much risk we are willing to accept. So this plot is from our data. What you see in the x-axis is the alpha level that increases, and then what we see here is the proportion of individuals. So we see that as more alphas, lots of individuals do have two labels, which is the blue, and then few of them only have one label, ME or non-ME. When we increase alpha, we allow the model to make more errors, so it becomes more decisive and more individuals tend to have only one label. And now we will discuss what the greens one means. So here, for instance, when we look at an example at an alpha 0.001. Let's suppose we have a new individual A arrives, we measure their blood biomarkers and NMR metabolites, and at alpha 0.1, the prediction set returned by the model is ME/CFS. This means that given the observed biomarkers, only ME/CFS is consistent with the model. So the algorithm can confidently predict ME/CFS status from blood biomarkers data alone. But now we consider another individual, B, for whom we take the same measurements, and at the same level of alpha, 0.1, the prediction set is ME/CFS or non-ME/CFS. In this case, the biomarkers are compatible with both outcomes, and the algorithm explicitly communicates that it cannot reliably distinguish between them. In some cases, as you can see here in the end in green, the prediction set can even be empty, and an empty set means that the individual looks unlike anything the model has seen during training, and it abstains from making a decision. So importantly, this is not a statement on whether someone has ME/CFS. It's a statement about what can and cannot be predicted from blood-based biomarkers and NMR measurements. So taken together, this plot is implicitly telling us that some individuals are easy for the model to predict, while others are more difficult. So the way to calculate how easy or how difficult a patient is to identify, we define credibility, which measures how stable a prediction is when we vary alpha. So for instance, here what we see in the plot is 3 individuals, A, B, and C. At the x-axis, we again have the alpha that goes increasing, and on the y-axis, we have the prediction set. So for instance, for individual A, the red one, we see that even at very small alphas, so with high credibility, the model is able to give that individual a single label, and that is kept throughout all the alpha range. Where for these other 2 patients, we see that at different levels of alpha, they switch from having 2 labels to 1, and from 1 to being empty. So this patient A, this individual A, what we're going to say is that it has high credibility because the width of the alphas that only have 1 label is very large, whereas the other ones are smaller. So basically, high credibility means the same level is predicted across a wide alpha range, while low credibility means that the prediction changes quickly as alpha varies. So before looking at the results, I'll briefly explain what SHAP values are. SHAP is a method for explaining model predictions at the level of individual participants. It's often used for explainability of machine learning models. And for a given individual, SHAP values quantify how much each feature pushes the model's prediction toward or away from ME relative to an average prediction. So importantly, SHAP values describe how the model is using the data. They are not causal effects, and they don't imply biological mechanisms. With that in mind, we can now look at how feature attribution relates to prediction confidence, if it does. So first of all, I wanted to show this plot. This is a very large plot. It's very difficult to read. But when I was mentioning that SHAP values pushes predictions towards or away from ME/CFS, what I was meaning is here in green are the patients who actually have ME/CFS, and in blue the ones that not. And what we see for the top features, which are the ones that in average contribute the more to the output, most green dots are on the right, meaning that it pushes towards ME, and blue are on the left, meaning that it's pushing away from ME. For the next plots, where we're going to analyze in more detail, we're going to focus on the first 6 features. And so, for instance, here we explore how feature attribution behave across predictions with different levels of conformal credibility. So each point is an individual. In this case, those individuals correspond to the top true positives. So individuals that did have ME/CFS, that the model predicted that had ME/CFS with the higher credibility. So with the largest width of alpha values. And it's only a way of saying that we see that high and low credibility predictions span broadly and similar ranges of SHAP values. So we do not interpret at this stage these patterns as evidence that particular features drive prediction confidence. Instead, this figure helps us assess whether uncertainty is linked to fundamentally different feature usage or whether the same features are reused with varying strength across individuals. So to better understand what this does and it does not tell us, we next asked whether these different prediction outcomes, such as true positive, false positives, and false negatives, rely on different features or show distinct attribution patterns. So here, this figure compares SHAP values for the true positive and false positive predictions. And what we observe is that the same features tend to appear across outcomes with broadly similar distribution of SHAP values. So in other words, incorrect predictions, and it would be false positives, are not characterized by different biomarkers. So it seems like the model is getting the same information. Based on the same information, it gives the same weight, and then it gives the same prediction that it would give to a true positive. So this suggests that prediction errors are not driven by distinct signals, but rather an overlap or noise or borderline cases within the current feature space. So given the similarity, so what we try to see here is how features varied across true positive, true negatives, false positives, and false negatives. So because we wanted to see if actually the underlying biomarker values that we were given to those features that the model deemed as very important were actually very different. I think here maybe the most interesting plot is this glycoprotein acetyls, which is the feature that our model now uses the most, or like it gives the most weight to drive prediction towards ME or away from ME. And what we see is that there is a clear overlap between the orange and the green curve, which are true positives and false positives, and then the other distribution corresponds to false negatives and true negatives. So basically what we are saying is that the features were similar, so it makes sense that the model labeled them the same. However, the model still makes different predictions across, and we see that the other biomarkers are not that different across the different populations. So that means those little signals also matter, and we need to understand how these signals relate to uncertainty and whether this actually means different patterns of any CFS in those borderline cases. So at this point, we've seen that model uncertainty does not arise because different prediction outcomes rely on completely different set of biomarkers. We also say that we also saw that predictions are not driven by a small offset of these things or obviously different features. And however, that does not mean that all predictions are equally reliable. What varies substantially is prediction stability at the individual level. By prediction stability, we remember, I mean, how an individual conformal prediction set changes as we vary alpha. Some individuals do it rapidly, while others remain at a single label. So the key point is that uncertainty here is not feature specific, but individual specific. And that's what motivates us to think about what this uncertainty may be telling us biologically. So this is the hypothesis. We see several plausible explanations for persistent predictions instability. First, ME/CFS is likely influenced by temporal dynamics that are not captured by a single plot draw. Second, there may be biologically distinct subgroups whose biomarkers signatures overlap in the measured space. One possibility is that uncertainty itself is structures. Different patterns of uncertainty may reflect different underlying ME/CFS subtypes, even when biomarkers overlap. And finally, it is possible that the key biological pathways relevant to ME/CFS are not fully represented by the biomarkers we are now using in the UK Biobank. So these are hypotheses that follow naturally from the observed uncertainty patterns rather than conclusions we draw here. So to summarize, conformal predictions allows us to move beyond point predictions and explicitly represent uncertainty at the individual level. In a heterogeneous condition like ME/CFS, this is particularly important. Uncertainty helps us identify cases where predictors should not be trusted and may also point to missing biological structure. And rather than forcing a decision for every individual, uncertainty-aware approaches provide a more honest and potentially more informative framework. So yeah, thank you, everyone. Here's my acknowledgement slide to everyone who participated in the study and gave their point of view. Also the funding, the PPI participants, and the scheme directors. And thank you, all of you, for listening, and happy to take any questions.
Thank you, Maria, for the great talk. There's a question by Nuno in the chat. Thanks for the presentation. For the model training, was the outcome data balanced? And also, are the model predictions fair in terms of age and gender, for example?
Yeah, so probably in one slide I mentioned what is the proportion of cases and controls we have. We see that there are 10 times more controls than cases. So what we do is we downsample the controls to the same number of cases, and we do it stratifying by gender, because as we know, ME/CFS is gender biased. Many more women are affected by the disease than men. And then in terms of age, this is not explicitly done in our cases control selection, but this is because UK Biobank tends to cover a quite narrow range of age, which is representative for both cases and controls.
Great. Thank you very much. So based on sort of what you're seeing, it seems that you're saying that you've reached a cap on average on how accurate a model can be with respect to the current available biomarkers, at least those in the UK Biobank. If you had, hypothetically infinite funding, what would be the kind of data you would generate to see if diagnostic accuracy can be increased?
So something, for instance, that is now quite a hot topic in the literature and we would be interested to explore is, for instance, proteomics. UK Biobank does have proteomics, but not for as many individuals as we would like. So that's something we would like to examine as well. And then obviously there is everything related to expression profiles or genetics, which happens to be with RNA or DNA, which are other types of biomarkers that could be informative, and we currently lack of the numbers that we need for those kinds of studies.
Thank you. Okay. If I'm not seeing any further questions in the chat, if there is anything further, please drop your question in the chat and Maria will be able to respond. Thank you very much, Maria. So the next speaker is Nuno from Warsaw University of Technology, with more than 20 years of experience working on applied statistics, genetics, genomics, epidemiology, and public health of infectious diseases. And he also has a series of work on ME/CFS. So the floor is yours. Thank you.
Yeah, can you hear me?
We can hear you. You were sharing and now it disappeared.
Yeah, okay, that's fine. And now you can see my presentation.
Yes.
And now full screen.
Perfect. Thank you.
Okay.
Yeah, perfect.
Good.
Thank you.
First of all, thank you for inviting me. It's been a great workshop, and I'm very happy to see a lot of quantitative biologists or data scientists being interested in ME/CFS and long COVID. Okay, so my presentation is slightly different from the ones from the previous ones. I'm mostly in this talk, I'm mostly interested in epidemiology, mostly on the estimating prevalence of ME/CFS in general. But given the COVID-19 pandemics and the onset of long COVID, I'm going to dwell a little bit on what we can do about estimating the prevalence of ME/CFS within the long COVID population. So that's actually the main question that I would like to address today. So first is what's the prevalence or incidence of ME/CFS in the population? We have been struggling with this question for many, many years. I was part of the Euromin consortium, and we tried to do a systematic review. And from what I've read in the literature, most people cite this systematic review and meta-analysis of prevalence of chronic fatigue syndrome. It has one of the very high number of citations, and this citation is actually in the 95th percentile of the Scopus database. But unfortunately, this systematic review has some problems, and one of them is just for you, just very briefly, is that when actually they screened the literature and then they found, like, according to their criteria, so they have 46 studies contributing to this systematic review and meta-analysis. But actually, when they actually did the analysis, they had like 56 prevalence data points. So they are putting more data than actually they should. So in this systematic review, the average prevalence of ME/CFS was around 1.4%. The aggregated prevalence across all the studies was less, 1.4%. And when actually they estimated everything by meta-analysis, actually it was 0.7. So it's around 680 cases per 100,000 individuals in the population, taking into account this. But the problem is that, and this is mostly also something that you also need to think about when actually you analyze this kind of data. I didn't want to ask a lot of questions about this, but when you analyze data, the data actually encompasses multiple case definitions, for example, for ME/CFS, and in long COVID, it's also dependent on that. So this data systematic review has this problem. You're using heterogeneous data, and sometimes sometimes it's actually due to case definitions. The systematic review has also a problem of a very long time period of papers that actually included in the systematic review. It was from 1990 to 2018, and this is one of the things that you also need to think about, is that inclusion of the studies actually do not represent the whole population, okay? And sometimes they screen just only adolescents. So when you analyze data, you should try to get data that is representative of the population, patients and non-patients. And of course, there was like a duplicated data, as I already mentioned. So the prevalence estimates that actually people are been citing in the literature, for me, they are not statistically sound and they should be carefully cited. So we actually try to do a little bit better than that. So I'm just going through that very briefly. And we did this under the EuroMIN network, so European network on chronic fatigue syndrome. And we actually, with our criteria, only 2 studies comply with our criteria, mostly on a random sample with good case definitions. So we focus mostly on 94 CDC case definition, and we focus on this study from England and another one from Iceland. And the prevalence estimates were around, in England, 0.2% and 0.1%, depending on the case definition. And in Iceland was around 1.4%, okay? So then it came the COVID-19 pandemics, and then people got interested in long COVID. And because of that, people also got interested in ME/CFS. So it's a little bit like the history of that. So when actually you start seeing, the first COVID cases, people thought immediately that this could lead to some kind of like post-infection sequela, okay? And so that's one of the things that, so in this situation, you actually had an opportunity to try to estimate prevalence of ME/CFS within the long COVID patients. Unfortunately, there's like just a few epidemiological studies doing that. And for this talk, I'm just going to give you like 3 studies, 3 of them conducted in USA and published, I think, in 2024. Okay, and depending on the sampling, so you have like in ones that was across the states, 8 sites, you actually like the prevalence of ME/CFS after SARS-CoV-2 infection is 2.8. And then if you go to other populations, like in California, it's around 2.2%. And this is a database of health insurance consortium called Kaiser Permanente. And I heard that people in Nima was presenting something about the Recover Adult Sponsor Initiative, and actually you have a paper that published around I think that last year, and it report the prevalence of 4.5%. Okay. Then people start again trying to compile data without very strong criteria. And this was published into the end of 2024, and this tried to estimate this prevalence of ME/CFS after SARS-CoV-2 infection. And there was 13 studies. They estimate 51% of the long COVID population having ME/CFS. But me and a friend of mine, a colleague from Austria, called Francisco Westermeier, we went through this paper, and actually the things that I was telling you about mostly is that the studies included in this meta-analysis, they actually don't represent the population. So they are biased studies, so they have like bias estimates. Okay, so we are still on, more or less in a starting point, and that's exactly the objective, a little bit what we can do about this. What is the prevalence of ME/CFS among long COVID? And now, given that you have some kind of like data available, and so I'm bringing you here a paper that was, I don't remember exactly when it was published, but it's maybe 2 or 3 years ago. It was exactly this Recover Adult initiative, and they were talking about how to define long COVID, or in their terminology, post-acute sequelae of SARS-CoV-2. Okay, so it's called PASC. And actually, they reported this kind of data, which is, I don't know if you can see it. The screenshot is maybe not in the high resolution, but actually this is like a data table. What actually you have on each row, the different symptoms. Each column represents different populations. One that is actually the whole overall population of this recovered adults. Then you have some individuals that actually were considered long COVID positive or PASC positive. Okay, and then you have subgroups within that. Okay, and the numbers that you're seeing there is actually the prevalence of each symptom. Okay, in these different populations. Okay, so I thought, why not to use this data to try to see if actually can estimate ME/CFS on these different populations. So this was proposed to a master student of mine in Warsaw University of Technology. So Patricia Jacques accepted this challenge, and she wrote a thesis about this, and she's going to defend in the end of the month. So the idea now is coming back to the definition of ME/CFS that I think it's a very important point to discuss. And mostly it's like CDC, the CDC 94 criteria, or Fukuda criteria, is you need to have like the patient should have an explained fatigue that persists more than 6 months. And when it says unexplained, it means that it's actually not explained by any disease that you know. And then you should have at least 4 of 8 core symptoms. And I just put here new headaches, tender lymph nodes, post-exertional malaise, sore throat, muscle pain, multi-joint pain, unrefreshing sleep, impaired short-term memory and concentration problems. Okay, so this is the case definition. So what the first thing that we have to do is actually to see if you have these symptoms in this like database. Okay, and then you have mostly the symptom that actually is more represented in this population is actually fatigue. And then you have 7 of these core symptoms related to the 94 CDC criteria. Okay, unfortunately, for this database, there was no information about tender lymph nodes. Okay. There's some assumptions that we did. So, for example, in this database, there was a symptom called brain fog. We actually considered this as concentration problems. Sleep disturbance, we considered as unrefreshing sleep. Okay, so these are some limitations on this straight away. So this is the summary of that data, of this big table now, but just related to the core symptoms that you're trying to model in a way. So you try the statistical question is that how to use this data to estimate the prevalence of ME/CFS in each these subpopulations. Okay, as you can see, fatigue is on the top. Okay, the most prevalent symptom, and then you have post-exertion malaise as the second. So what actually we thought is to use a method that is actually used a lot in surveys, and it's called the racking method. So mostly is what we're going to do is based on several steps that I'm going through. So mostly is to try to bootstrap the symptom profiles of the individuals, then you apply ranking, you do the prevalence estimates, you repeat this process, coming back to bootstrapping of your sample, and then you get a set of estimates, and then in the end you can calculate like a mean prevalence. Okay. So the idea of the first step is actually you generate a sample of n profiles with replacement from all possible symptoms that you can get in the individuals. Okay. So in this case, we only have 7 core symptoms plus fatigue. So if you make this all possible combinations, you have 266 profiles. That is represented by this set P. Okay, and then you take a sample, random sample from there. Okay, and this is, for example, is the profile 54, profile 11, and profile 154. Okay, and then you compose this as a sample. And now what you're going to do, you're going to apply the racking. And what the racking is doing is that given that you have a certain symptoms profile in the sample, you're going to give a weight to each of the proportions that actually related to the original marginal prevalence of the symptoms. Okay, this is mostly what I'm going to show you next. So what actually we're doing here, when you actually have this sample and you have like a symptoms profile for each individual, we're going to give a weight to each individual that overall, when actually you calculate the prevalence, you're going to match that prevalence with the prevalence that actually it was on the original data sets, where actually you don't have the raw data. Okay, so this is actually has to be done numerically. So it's like you actually have to solve this as this kind of like set of equations. Okay, and there's our package that actually can do that. Is called IPFR. Okay, so you get the solution of the weights, and then after having the weights, then you can calculate the weights that are complying with the symptoms or the symptoms profiles that actually comply with the case definition. Okay, that's what is represented here. So the prevalence, according to the CDC criteria, is the sum of all the weights of each profile that you generated by the bootstrap, and then you just sum and you get the prevalence estimates for that sample. The thing is that then you have to repeat this process several times, let's say 1,000 times, and then you can pull them for each time you're going to have a prevalence estimate. So you just take an average. If you want to calculate the uncertainty, you can calculate the quantiles 2.5% or 97%. Okay. So if by this just basic algorithm, you can have so for the overall population, so all the recovered data set that is almost like 10,000 individuals, the prevalence of ME/CFS is around 0.3%. When you go to the PASC negative, unspecified, okay, so you don't know, so the sample size is around 7,700, and actually the prevalence is very, very low, and it's below 0.1%. For the PASC positive, okay, which is around 2,000 individuals, you get 32%, okay? So for each one of the clusters that are there, you're going to see that actually there are gradients on the clusters. So there is 2 clusters that actually are related to low prevalence of ME/CFS, and then you have other 2 clusters that actually they are much heavy in the prevalence of ME/CFS. So with the last cluster, that actually the people have a lot of symptoms. You have the prediction of these methods. It gives you around 77% of the patients have, or they should comply with ME/CFS case definition. Okay. So the problem is that I didn't mention this, but this method is going, actually, you are just using the aggregated data. And actually, when you just don't put information to get any additional information, what you're going to get is to estimate the prevalence is actually to multiply to get the probability of a given profile. You just multiply the probabilities of each symptom. That's the solution that the algorithm is going to give you. And it's not for us, it's not so informative. And so we thought about something that is more actually a variation of this method that is related to actually we know about the disease. So, for example, for ME/CFS. And what we did is that among this set of this 256 symptoms profile, you're going to exclude a few of them according to the number of symptoms that you have, that the profile has, given that that symptom profile doesn't have fatigue, okay? So when people have their absence of fatigue, they shouldn't have a lot of symptoms, okay? Because fatigue will influence, in a way, other symptoms as well. So we fix the number, you remove from these symptoms profiles some these symptom profiles that actually have a lot of symptoms. So you can change the number of symptoms that we are going to exclude by this parameter kappa, and you try and you see you do like a sensitivity analysis when actually you change this kappa, from removing all non-fatigue symptom profiles with one more than one symptom. And you do that until you remove remove the individuals that actually have all the symptoms, given that you don't have fatigue. Okay. So this is kind of like a sensitivity analysis. So by removing some profiles, just all the profiles, all the profiles that actually have all the symptoms, is like this number 7, okay? Is the KPK7. So it's very close to what you got before, but actually when you start truncating this kind of this symptoms profile set, you start increasing the probability of the prevalence of ME/CFS among these different populations. So in this case, it's particular to the PASC population, okay? So you can see that in the most, let's say the worst case scenario, the prevalence of ME/CFS among PASC is going to be around almost like 50%, okay? You can do the same thing. I'm not going to bother you with all the results, but this is for cluster 1 and cluster 2, okay? Here in this case, you don't have a lot of ME/CFS patients, but in these groups, one can go, in cluster 1, it can go up to more than 20%, and the other cluster 2, you can have slightly less than 15%. For the remaining ones, actually, it's much higher, but in cluster 4, and that's mostly, is like in the worst case scenario, you're going to have like prevalence almost 85%. Okay, so the take-home message of this talk is that we can try to estimate the prevalence of ME/CFS with symptoms data that actually are dispersed in the literature, using this kind of like ranking method, and you try to plug in some kind of information, about the symptoms, because that not going to have like symptoms profiles that are not going to be observable. So you can try to see what's kind of like the sensitivity analysis that you can get in terms of this prevalence estimates. So the final prevalence estimate is depending on this how much you're going to truncate of this symptoms profile set. My proposition is around 4, which is related to the Fukuda, okay? Because you not expect, people in absence of fatigue to have a lot of other symptoms, okay? So you should have around 35% among the PASC positive. Okay, and this is our acknowledgement. I'm very happy to take questions, and I'm sorry if I extended, but I extended my time, but I cannot see the clock in my computer. Thank you.
Thank you very much for the great talk. There's a question from Chris in the chat. Is estimating ME/CFS prevalence amongst those with long COVID using Fukuda criteria useful? That's the first question.
Useful?
Yes.
Well, one thing is that, and this, I must say, this is, I'm Portuguese, so, and I'm saying this also from the point of view of public health, okay?
Right. So I should have also it says, as you say, the estimate will include some people who do not have PEM. I think that is why he's asking the question. So why not use?
Yes, you're absolutely right. Actually the Aeromean network, and when actually they proposed like the new guidelines for disease diagnostics and treatment, we came to a consensus that actually you should have fatigue plus PEM, and then the Fukuda will modify it accordingly. But this is just for illustrative purposes. So I agree with Chris. Actually, we should have the two things together. So that actually then it's much easier to let's say to comply with actually what people consider nowadays ME/CFS, which is mostly fatigue plus post-exertion malaise and neurological and autonomic symptoms. Yeah. If I want, I just I don't know if Chris asked me why to estimate the prevalence of ME/CFS among long COVID, and when I wanted to ask that question is that this is very important from public health point of view, because here in Portugal, people don't recognize, they recognize long COVID, but ME/CFS is like absent. Most people are diagnosed with fibromyalgia, and actually they have ME/CFS. So we are trying to find a way to convince authorities that actually ME/CFS is not only, just something, from the past, is actually related to the present when actually you have a long COVID and a big chunk is there. Okay. So that was the main purpose, to find some kind of prevalent estimation so that we can try to convince the public health authorities that actually this is a real problem. And because without statistics, we cannot work and convince them that we should have more funding or so. Okay.
Excellent. Well, thank you very much. And yeah, I guess we're ready for the concluding remarks. So I can try to share. Yeah, so basically we let me just go back one step. So thank you very much for all of our speakers. We appreciate that sort of this was a quantitative series of talks, but we wanted to have some exposure about the various kinds of methodologies people are building in this space with both long COVID and ME/CFS. And some of the key considerations that were discussed multiple times is clarity about what the scientific question of interest is, what kind of data we're looking with, and how does the structure of that sampling affect how and whether or not we can answer the scientific questions of interest, or we have to limit it to a kind of sub-question. We talked a lot about rigor, so choice of methodology, what assumptions do they have, and is the application of a certain method to the given data is actually going to be valid. This should not be underestimated because this can really change our conclusions about what we do, especially in translational topics such as ME and long COVID. Finally, we also spoke a lot in a variety of talks about uncertainty quantification. We're not really coming up with a single number to make a statement, but how we rigorously come up with our uncertainty about the results, and how do we in fact communicate that to the patient and public community. And you will have also noticed that in a series of talks we try to separate off, well, actually last talk was on prevalence, and then estimation problem, population statements versus individual level predictions, and the assumptions that go into each are different by nature, and the methodologies are also relatively different by nature. Okay, so that's a summary, a short summary of some of the considerations. And the final thing to state is that the next Prime workshop will be on the theme of the similarities and distinctions between long COVID and ME/CFS. The provisional date is going to be on the 24th of April, 2026, between 2 to 5 p.m. UK time, and the registration is expected to open towards the end of February. And of course, as Chris also mentioned at the beginning, this is our first workshop of this kind. So if you have any feedback or suggestion, please do reach out. Furthermore, if you have any questions for any of the speakers, or if you have ideas for potential collaborations with any of the speakers, that would also be most welcome as well. Thank you. Great. Thank you all. Bye-bye.